Dear students, welcome back to embryology classes. And as we know, the embryology classes are continuous sessions. So it is good to know continuously as a whole part. Then only we will have a better idea about every topic. So in previous sessions, we had completed about spermatogenesis and oogenesis, where we saw the development of female pronuclei and male pronuclei, which fuse together to form zygote. So today's step is about fertilization. So if you see the development under general embryology, it is like first week, second week, third week and fourth week. In first week, we are going to learn about fertilization and implantation. And in second week, it is going with formation of bilaminar germ disc. And third week, we would look upon the trilaminar germ disc, that is the formation of 3 germ layers called as gastrulation. And finally, the fourth week, where we will see the foldings of embryo and further development. And further, we would continue with the systemic embryology, where all the different organs and systems will develop. So today's session is on first week, that is fertilization. So fertilization, at the end of spermatogenesis, we have got a matured spermatozoa, and at the end of oogenesis, there will be a oocyte, that is oocyte in the secondary oocyte. So the stage which we see here is secondary oocyte. So here the secondary oocyte, it is the cell division in the oocyte, it is arrested in meiosis 2. So second phase of meiosis. To be more specific, it is in the metaphase. So during metaphase stage, the secondary oocyte is arrested. So the fate of secondary oocyte depends upon fertilization. Suppose the secondary oocyte undergoes fertilization, means it fuses with the male gamete, then it divides, it completes this meiosis 2, and finally it forms a matured ova. If the fertilization doesn't occur, then the secondary oocyte will be remain in the same stage and it will get perished off, and it is removed from the uterus during menstrual cycle, that is during menstruation in women. Here is the picture showing the uterine cavity. So this is the uterine cavity, and these yellow colored structures are the sperms which are entering into the female genital tract. And uterine cavity, on each side they communicate with the uterine tube, which are called as fallopian tube. And fallopian tube further divided into isthmus. Isthmus is the constricted part of the fallopian tube. Dilated part of the fallopian tube is ampulla of the fallopian tube, and this finger-like projections is called as fimbriated end. Fimbria means finger-like, where it is related to ovary. And the part of the fallopian tube which is present within the wall of the uterus, it is called as intramural part. So these are the parts of the fallopian tube. So where the fertilization occurs? The fertilization usually occurs here in the ampulla of the uterine tube. To be more specific, it is at the junction of isthmus and ampulla. So to say, it is ampulo-isthmus junction. So at the junction of isthmus and the ampulla is the normal site of fertilization. Suppose the fertilization occurs elsewhere, it may result in abnormal, that is ectopic pregnancies. So that we would discuss little later. So this is normal site of fertilization. So now the time taken for this process of fertilization is 24 hours. Approximately around 24 hours fertilization process will get over. So if you see the number of sperms, usually they get deposited in the vagina of the female tract. They are around 200 to 300 million sperms. So out of these 200 to 300 million sperms which normally deposit in the female genital tract, only 300 to 500 reach the site of fertilization, that is to the ampulla. So how many reach the ampulla? Only out of this 300 to 500 sperms. So out of this 300 to 500 sperms, only one of these will have a chance to fertilize the ovum to form a zygote. So this is the competition, how the sperms reach to the sperm. So this is how they travel from the vaginal tract to the ampulla of the uterine tube. So before undergoing fertilization, the sperm has to undergo some pre-fertilization changes. So this pre-fertilization changes includes a main step called as capacitation. So only the capacitated sperm is allowed to undergo fertilization. So capacitation, it is a period of conditioning of the sperms in the uterine tract and which lasts for around 7 hours. So if you see the structure of the sperm, it has got head, a constriction which is called as the neck, and neck continues to form the body and body continues to form the tail. This is how a sperm will be, and the head, it is covered by a cap. This cap is called as acrosomal cap. So the acrosomal cap contains some specific proteins which are called as glycoproteins and seminal plasma proteins. So these glycoproteins and seminal plasma proteins are to be removed before undergoing penetration. So during capacitation, it is the period of conditioning of the sperms within the uterine tract where the acrosomal cap, that is the proteins present in the acrosomal cap which are called as glycoproteins and also seminal plasma proteins. So these proteins has to be disintegrated and removed within the uterine tract. So removal of this acrosomal cap is called as capacitation. So let's see about the penetration. Penetration occurs in 3 stages. One is penetration through corona radiata, second stage is penetration through the zona pellucida, and third stage where we see the fusion of oocyte and the sperm cell membranes. So what is this corona radiata? Corona radiata you will know better if you know the oogenesis. So here if this is a normal oocyte, so normal oocyte is surrounded by a layer of a glycoprotein layer here which is called as zona pellucida. I'll zoom it for you all. Corona radiata are this group of cells which are surrounding the oocyte. So corona radiata is nothing but the group of cells which are called as cumulus ovaricus. They are present around the oocyte, which are the follicular cells. So during the development of Graafian follicle, some follicular cells surround this primary oocyte and that is the secondary oocyte for the division. So this group of cells together called as corona radiata because it is like a crown surrounding around this secondary oocyte. So the capacitated sperm, we know already what is capacitation. So that is the removal of proteins from the acrosomal cap is capacitation. Only the capacitated sperm can enter. So out of several sperms, only one sperm can enter through the corona radiata fully to touch the zona pellucida. So rest of the sperms which are not fertilized in the egg, so they just aid one sperm to enter inside. So that is the function or action of the other sperms. So only one sperm enters inside. And second phase is the penetration of the sperm through zona pellucida. We know it now. The sperm touches the zona pellucida layer, which is, zona means a layer, pellucida means a translucent layer. So it is a translucent glycoprotein shell surrounding the egg, and that facilitates and maintains the sperm binding, and because of this it induces a acrosome reaction. So what is this acrosome reaction? This reaction is initiated after binding of sperm to the zona pellucida. Only when sperm comes in contact with the zona pellucida, this reaction will begin. So this reaction is induced by a special, this reaction is induced zonal proteins. So zonal proteins, in short we'll say ZP3 proteins, it is represented as ZP3, and which releases enzymes like acrosin and trypsin. So these zonal proteins releases enzymes called as acrosin and trypsin. So this acrosin and trypsin, it allows the sperm to penetrate the zona. So that is the function, and also the acrosomal cap is now completely removed here. Only the sperm is allowed to penetrate through the zona. Let's see about the third stage. The third stage, the oocyte and the sperm cell membranes, here they fuse, that is the fusion of the cell membranes is the third stage. The cell membrane of the oocyte and sperm fuse here. The head and tail of the sperm enter the cytoplasm of the oocyte, and the cell membrane and the mitochondria of the sperm, it remains in perivitelline space. So the space here between the zona pellucida and the cell membrane, this blue color area here, this is perivitelline space. So the perivitelline space is this gap between the oocyte, the cell membrane of the oocyte and the zona pellucida, within which we can find first and second polar bodies. So the matured ova is ready now, and now the male pronucleus enters into the cytoplasm of the female oocyte here. So these two are the male and female pronuclei, which contains haploid number of chromosomes each, that is 23X or Y, 23X in females. So these two pronuclei, they fuse to form a complete zygote having diploid number of chromosomes, that is 46 if it is female, XX, 46 XY, which is male. So determination of the sex happens at the end of the fertilization itself. So we saw the second meiotic division is completed now. So the second polar body also settles in here in the perivitelline space, and pronucleus, female pronucleus meets with the male pronucleus to form a single nucleus. So before it forms a complete cell, single cell itself, the cell starts undergoing cleavage, that is starts undergoing differentiating further into 2-celled stage, 4-celled stage, 16-celled stage. So that we will discuss little later. So it is not a complete single cell actually. It just differentiates into group of cells immediately. So here the chromosomes are formed and breakdown of nuclear membranes occurs. So complete pronuclei, and here the cleavage spindles are formed again, and the formation of the zygote. The chromosomes of the zygote get arranged in the equatorial plate where it enters into cleavage, means cell division. So this is the different phases of fertilization. So first is phase, to be in short, penetration. First phase is the pre-fertilization changes, that is capacitation of the sperm, and second phase we saw the penetration. So penetration is through corona radiata, second penetration is through zona pellucida, then third stage fusion of the cell membranes of oocyte and sperms. After fusion we have seen the formation of male and female pronuclei, and here the replication of DNA occurs, and now the chromosomes, they form into a diploid number, that is 46XX or 46XY. Then finally the zygote is formed. Okay, let's see the results of this fertilization. One thing is, it stimulates the penetrated oocyte to compute the second meiotic division is completed because of penetration of the sperm. So penetrated oocyte will complete the second meiotic division. That is first result. Second is, restores the normal diploid number of chromosomes. Each gamete, that is male and female gamete, will have haploid number of chromosomes. So it restores the normal diploid number of chromosomes, that is 46 in the zygote, and determines the chromosomal sex of the embryo. So whether it is male or female, we will get to know in the beginning because of the formation of zygote. If it is 46XY, then it is a male zygote. If the chromosomes are having 46XX, then it is a female chromosome. And this zygote, as I told you all, before it forms a complete cell, immediately it initiates cleavage, that is cell division. So cell division also initiated at the end of fertilization. So let's see some clinical aspects of this. Infertility is the problem of 15 to 30% of the couples and can overcome using assisted reproductive technology. So what is this assisted reproductive technologies? Like in vitro fertilization. So where it involves the fertilizing the eggs in a culture medium and placing them back into the uterus when the cell divides to 8-cell stage. So when the zygote divides to form 8-celled stage, at that stage they will put back the morula, that is 8-celled stage structure, into the uterine cavity. That is called as in vitro fertilization. Fertilization occurs outside. Vitro means not in the inside, not naturally. It occurs artificially. Second assisted reproductive technology is intracytoplasmic sperm injection. That is fertilization of ovum by injecting that by intracytoplasmic sperm injection, where by a single sperm injected into the X cytoplasm. So one sperm is taken and it is injected directly into the egg when the woman is about to do ovulation. So before entering into the ovulation phase, at the time of ovulation, they do this procedure. This is called as intracytoplasmic sperm injection. So I'm writing short form, intracytoplasmic sperm injection. And in vitro techniques are often associated with increased risk of birth defects. So that is one risk of problem in having in vitro fertilization. And also not only birth defects, prematurity, low birth weight, and multiple births like twins, triplets or quadruplets. So multiple births are common with in vitro fertilization. So this completes a little clinical aspect and fertilization with different phases. Thank you.